Can you all see my
Mhm.
screen?
You're good to go.
Perfect, thank you. Yep, and as Dan said, guys, this is my work from my PhD, and thank you all for joining us for this talk. And I performed it in Axel Kali's lab. And for my PhD, I really had the question about really who were the critical players in the immune checkpoint response. And so we really knew CD8 T cells were important. We knew that there were certain subsets that were coming up in patients that showing to be maybe correlated with better survival, better responses, but we really weren't sure biologically, functionally, who was the most important. And that's really where my project came in. And so first up, why do we look at T cells or CD8 T cells? Like I mentioned, these cells can directly eliminate tumor cells, and they are the main responders to immunotherapy, or such as immune checkpoint blockade, which I'll be calling ICB here. However, within the tumor, we have chronic antigen exposure. We have a very immunosuppressive tumor microenvironment, and this causes all of the majority of effector CD8 cytotoxic T cells that could kill tumors to really become dysfunctional. And we like to call these cells exhausted. They express markers such as PD-1, and these exhausted T cells are very much worse at killing. So they have now inhibitory receptors on their surface. They have reduced cytokine production, which result in tumor cell death, and they also proliferate much less. And this all leads to them not being able to kill tumor cells very efficiently. But we know that this T cell pool is very heterogeneous, and I'm just going to take you through a few states, especially the states that are important in that checkpoint blockade response. So first up, we have precursors of these exhausted T cells, and we call them TPEX. And these cells are marked by the ability to express not just toxin exhausted molecule, but TCF1, which really helps us identify these cells and self-renewing and stem light. And these cells are what differentiate and sustain the TX subset. So these TPEX cells are uniquely different transcriptomically, epigenetically, and also metabolically from other more terminally exhausted TX cells. I haven't touched on it here, but we also know there's further heterogeneity within this pool, which I will touch on later. A second set of, and of course, they expand after a checkpoint, which is really important. And they've also been noticed to be found not just in tumors, but also tumor draining lymph nodes of both patients and in tumor models of mice. So the second set that we know is tissue-resident memory cells. So these cells were first classically identified in acute infection, and they are non-recirculating, and some transcription factors that we use to identify them are Hobbit, RANX3, and BLIMP1 positive cells, and they're KLF2 negative. And surface markers are mostly integrins that we use to identify them, such as CD103, CD69, and CD49A. However, in tumors, because tumors are a tissue, so we could have cells that are residing in there showing these resident features. But because in a tumor, we still have antigen present, in TRM cells, it's a memory time point, we don't have antigen present. So because of this kind of mixed state, we call it a tissue resident memory like T cell, because yes, they have these phenotypic features of TRM cells, but they are present in an antigen environment, full environment. And why are these cells coming up now in cancer? Because the presence of cells marked by CD103 and CD69, two canonical markers of TRM cells, have been shown to correlate with improved patient survival. And interestingly, in tumor, they have been shown to also prevent tumor development and have a really important role in tumor surveillance. And we have also have been papers that have shown that these TRM-like cells do have cytotoxic function. And so now these cells have been identified, unlike the T-PEC cells, not in the tumor draining lymph node, but within the tumor tissue. And so, really, the key questions of this paper was, can we define T cell fate within the intratumoral T cell pool? Not just in the tumor, but in the lymph node. What cells can we find in tumors that also have these TRM-like states? And then we want to say, okay, we identify what cells are there. What was critical? Who was critical for driving this anti-tumor immunity? And I won't talk about it here in today's talk just because of time, but we also went into trying to uncover mechanisms within this tumor niche that can change the programs of T cell fate. So first up, to be able to identify what cells were in there, we did single cell RNA sequencing. So we chose the 83 ova subcutaneous tumor model. It is a breast cancer, and why we chose it because we did look at quite multiple tumor models. You will see that in the paper, but we found that the TRM-like cell population was highly found in this tumor. So while TRM-like cells can be found in abundance in quite a few epithelial human cancers, it is more rare in mouse cancers. So you have to be careful when picking your model. And then we give some mice checkpoint, but also left some of them untreated, and we sequence them, and we sequence active all the activated polyclonal cells from the tumor. But because the tumor draining lymph nodes also have lots of naive cells, to get antigen experience, TCR experience cells, we get on PD-1 positive CD8 T cells, and then the single cell. And as you can see here, we had quite, when we did a UMAP for all our cells, we had quite a few nice clusters. But what was really interesting was that the biggest separation of our cells was the organ. So here you can see that the cells over here are from the tumor. When we look at where the cells were, well, the cells on the right were all from the tumor draining lymph node, really showing that these cells, just by tissue, really can be mapped quite differently or separately. Then we were looking at what cells were really there. And then before we did that, we looked at the signatures. So first we were saying, okay, what type of cells are in there? So what we did was we went back into the literature and we picked some signatures from the literature. So first we picked a TPEX signature and a TX signature from the same paper, and also an TRM signature. So these are all from chronic infection or acute infection. So the classical signatures for these true cells. And you can see here that the TPEX cells, those precursor cells, were really found in the lymph node, those cells on the right, but also we had some subsets of TPEX-like cells within the tumor. You can see that these TX and TPEX signatures weren't co-expressed. It was either a TPEX cell or really a TX cell. So we had TX cells a lot in the tumor, but we did not have a high TX signature in lymph nodes. TRM signatures again were not high in the lymph node, but we could see TRM cells in the tumor. Interestingly, you can see that the TRM cell did overlap with some of the TX signature, and at the same time, the TRM signature also overlaps in different clusters with the TPEX signature, showing us that we had this overlap of the fate of TPEX, TX, and TRM cells. And interestingly, we also want to make sure that those TRM-like cells we were seeing in my mouse tumor were similar to those TRM-like signatures found in human tumors, those TRM-like cells that we were shown to be important in human cancers. And that was in melanoma and TNBC. In both these papers, they showed that there's a high correlation with having TRMs and better survival or better responses. And again, you can see that our TRM-like signatures overlap very well. TRM-like cells, as identified by the signature here, also identified well with the human TRM-like signatures, saying that we could find those TRM-like cells in our model as well, showing us that our model right here is also clinically relevant. And so what cells were we seeing in here? So it's just a quick breakdown of what we were seeing in our tumor and lymph nodes. First up, we could see here that we did have TRM-like TX cells. And why do I call it that? Because it had the TX and the TRM-like signature, and that was mostly cluster 9 over here. Then we also saw more conventional TX cells. When I say conventional, I mean non-TRM-like. And you can see that we have TX cells within the tumor, but we also had, even though it wasn't very high for a classical terminally differentiated TX signature in the lymph node, we had this cluster 3 that was CX3CR1 positive. And you all may know already that CX3CR1 has been shown to be expressed by certain TX cells that have shown to have the higher effective function. And next we had TRM-like TPEX cells, and that was in cluster 5 over here because it was high for the TPEX signature and the TRM-like signature. And we saw not just in the tumor, but in the lymph node, more conventional TPEX cells as well. Interestingly, if you look here, I have identified cluster 1 and 2 as cell negative or cell positive, which is CD62L. And this is because actually Carlson, who's talking later, identified in a paper recently, well, now a few years back, that you could actually see CD62L positive, MIB positive cells in the lymph node. And these cells were better, but they were the key responders to checkpoint therapy, that they gave rise to the burst. So we were interested to see that, yes, we could see those cells as well within our model. And so that's what we ended up having, this nice model where we could really identify TRM-like cells but also precursor T-PEC cells and TX cells. And now that we could identify these cells, we want to be able to actually manipulate them, see them via FACS and flow. So we were using in our lab ID3 to report a mouse to identify our T-PEC cells quite easily. And you can see here, even within my tumor, TCF1, the key mark of T-PEC cells, is co-expressed with ID3 almost completely. And then, if you remember, the transcription factor I mentioned for TRM-like cells was Hobbit, and we looked in acute infection and other tumor models where we confirmed that TRM cells within these models do express the marker Hobbit, and we could also see that Hobbit was co-expressed with more with the TRM marker, TRM-like cell markers from human cancers, such as CD103 and CD49A, helping. So we've in our model here used Hobbit to identify TRM-like cells. So now we had a way to look at T-PEC cells and a way to look at TRM-like cells, but I want to look at them at the same time. So we crossed these two report to my mouse, and we made a Hobbit tomatocree ID3 GFP mouse. And I'll just walk you through this. So you can see here that we have some cells, the reds and the blues, that express Hobbit, and we have some cells that express ID3, which is the greens and the blue. So what we have over here in the green are the ID3 single positives, and they were the lowest frequencies in most tumors. And we had a really nice big population of these blue cells that co-express Hobbit and ID3, and we identified these as TRM-like T-PEC cells because they are in the tumor tissue and they express these markers of both Hobbit and ID3. And then we had Hobbit single positive cells. So they didn't have any T-PEX markers, but just Hobbit. And then we had cells that were more double negative. I would like to just mention here quickly that we could find these cells also within tetramer positive cells. And if I looked at the tumor draining lymph node, non-draining lymph node, spleen, we really did not see Hobbit expression. And Hobbit was very much just seen within the tumor tissue. And so it was more of a tumor specific marker when it was upregulated. So now we were able to see these cells and we were able to just look at, I did a lot of phenotyping, but just to confirm that these ID3 expressing cells, the blues and the greens, they were high in TCF1. Very interestingly, it was only my double positive, so the TRM-like precursors, that expressed CD103. And this was interesting because this was the marker that was identified in patients to identify the TRM-like cells that was shown to be important or thought to be important in checkpoint inhibitor checkpoint responses. CD49A was also expressed in these horribly positive cells and some of the double negatives. And interesting, you can see here that TIM3, TOX, and PD1 were the most expressed on the Hobbit single positives and double negatives, marking them to us as TX cells. And that is why I would call those Hobbit single positive cells as TRM like TX cells, while the double negatives are more conventional TX cells. And just a few more markers, but I will just skip that for now. And again, you can see here the CD103 very high in the blue in my TRM like TPEX. But we also went back and did some bulk sequencing just to see those signatures again. And we could really prove, I mean, not prove, but show that this TX signature, as you can see here in red, is highest in my double negatives and my Hobbit single positives, while the TPEC signature was the highest in my ID3 positive populations, including my TRM like TPECs in the double positives and my ID3 single positives. And this heterogeneity was actually observed in multiple models. So the number of these, the proportions of these populations were different, but in multiple subcut models such as MC38, B16, and even orthotopic 83 over, I could identify these cells. And when I look at them, their phenotype, it was pretty much what we saw up here. So it was quite consistent. And so what we have identified right now is we were really focusing on the tumor at this point and trying to identify the cells within the tumor and trying to identify what precursor or T-PEC cells were found in in tumors. And so we confirmed that in tumor models, ID3 can also identify T-PEC cells. We found that Hobbit identifies TRM-like cells. Most interestingly, we were able to identify novel TRM-like T-PEC cells, and these were marked by CD103. And we saw that the Hobbit TRM-like cells and the Hobbit ID3 double negative cells were both terminally exhausted to the same extent, even though I didn't go too much detail here. It was more in the paper as well. And now that we identify the cells, we really wanted to be able to see who was driving what. So we went into more functional work. And to this, we have quite a few mouse models that I will go into later, the genetic. But we first started with my lovely double reporter, and I gave them checkpoint inhibition. And what I could really see was that my four populations were really reorganized after checkpoint. So these cells were differentiating and repopulating each other. And interestingly, we saw my double positive TRM-like cells disappear. And to us, that made us think maybe these cells were differentiating and these giving rise to effectors, and maybe they were really the important cell type to checkpoint. But interestingly, we saw that my Hobbit single positive, so those Hobbit TRM like TX cells, they didn't expand a lot. And I also did fate mapping mice. So I fate mapped the Hobbit lineage and then gave them a checkpoint. And I really noticed that the Hobbit lineage cells did not expand to the same extent as the Hobbit non-Hobbit cells. So really making us realize that it wasn't TRM-like lineage of precursors or effectors that were driving checkpoint in terms of numbers and infiltration, but the non-TRM-like cells. But we still want to see how important were they. So we made a Hobbit Cree TCF1 knockout mouse. So the Hobbit tomato mouse we had also had a Cree, and this meant that any cell that expressed Hobbit would knock out TCF. So the cell would still remain within the tumor, within the mouse, but will have no longer express TCF1, so they lose that TPEX identity. So as you can see here, we have Hobbit versus TCF1, we have those Hobbit expressing precursors and TX cells over here. But in my knockout mouse, we lose any Hobbit cell expressing TCF1, showing the model work. I did a lot of validation as well for this. And interestingly, we noticed that this did not impact any of the cell numbers, proportions, or any of the other immune cell types as well in numbers within the tumor during lymph node or spleen, mostly because we saw Hobbit being expressed in the tumor. And so this tracked with that. And so Hobbit really was being, or only my tumor-specific TRM-like precursors were not present anymore. Yep, and just to show you here again that it was, and these cells, because they were losing the expression of TCF1, they would now become more effector-like, so they would now differentiate, and that's why we had more TIM3 positive, activated granzyme B positive, more activated cells at steady state, because they have now, because without TCF1, they had to differentiate. But okay, without these TRM-like precursors, can we still have tumor control? That was our big question. And so we gave these mice checkpoint, and surprisingly, there was no difference. So not just at steady state, was there no difference in tumor growth, but these mice, even without having TRM like TPEX cells, were still able to control their tumors. And to us, this showed that these TPEX like TRM like cells played a limited role in tumor control. And there was just some numbers to prove that, because they could still mount that immune response even with TCF1 knocked out. To go for a more stringent approach, we went with the rollout 26 DTA, which means that any cell that expresses Hobbit starts to express the diphtheria toxin, which means all of these cells are deleted. So any cell that expresses Hobbit is deleted, which means there's no more TRM-like cells, TX, TPEX, any of them, no TRM expressing cell, a cell that expresses Hobbit will remain in this mouse. And you can see here in my DTA, we had no Hobbit expression. And so what we did again was get a checkpoint. Now, could they still respond without these cells? And surprising to us, they still could. So this really proved to us that these TRM-like cells are dispensable in mounting an ICB response. But we really want to make sure who is responding. These TRM-like cells are not responding, but they were to show that it was precursor TPEX cells. So we just as a proof of concept, we had CD8 Cree T7-1 flux mice, which means that any CD8 cell will lose their TPEX identity. And here we prove that yes, within our model, TPEX cells do play a role in checkpoint, because if you lose all TPEX cells, you will lose your ability to respond to checkpoint as well. And this proved to us that it was the non-TRM-like cells that were critical to checkpoint responses. As you can see here, with even with looking at survival of the mice, we had much worse survival when you knocked out all TPEX, the TPEX identity from CD8 T cells. And now we just want to be really sure what cell that was driving it. And when we went back into our single cell, we looked at what expanded and shared clone type existed because we also sequenced mice who received checkpoint. And what we saw that most of the clones that were expanding were in the tumor draining lymph node. And we didn't just see the tumor draining lymph node TPEX expanding, but also a population of CX3CR1 expressing effector-like cells. And so, as you can see over here, we had a much higher number of shared clones off a checkpoint. And like I said, highest in the TCX3CR1s. And we also did slingshot analysis to see about the differential trajectory of these cells that were possible. And again, it was showing us that it was from the tumor draining lymph node that our effectors, also cells coming to the tumor after checkpoint, were cells coming from checkpoint. So all of our analysis was leading us to the lymph nodes. Now we pivoted from the tumor in the lymph node, and most of the lymph node work was done by my brilliant colleague Lisa Rausch. And what we noticed that when we gave mice checkpoint, we had a massive expansion of PD-1 positive cells within the tumor draining lymph node. And in line with our sequencing data, we saw most of these cells that were expanding were CX3CR1. So we had a higher proportion of CX3CR1 positive cells in the lymph node. And you can see that they lose Ly108, another TPEX marker. So these TPEX cells are now differentiating into CX3CR1s. And just to prove that these cells were being formed in the lymph node, we also gave these mice FTY720, which blocks egress, which means any cell that was made in the lymph node was stuck there. So we could identify what cells were forming. And you can see over here that we had an expansion of these PD-1 positive CD8 T cells after blocking egress, showing us that PD-1 positive cells at steady state were being continuously being made in the lymph node. But most importantly, even at steady state, we are having a massive amount of CX3CR1 positive cells coming from this lymph node. But now we wanted to confirm it was these cells coming from the lymph node that were important for checkpoint. So what I did was give my checkpoint, but also give them FTY 720 and then give them checkpoint. So I was blocking egress from the lymph node in some mice and then give them checkpoint to compare that immune response. And most interestingly, if I block any egress from the lymph node, the mouse loses the ability to respond to checkpoint and they lose the ability to control their tumors. So finally, we were seeing the loss of tumor control, and this was just when we gave checkpoint after blocking. So it really showed us that you needed this continuous replenishment into the tumor to maintain the ICB response in our mice. And as you can see here, if you don't have this replenishment, you lose the total number of CD44 cells, of course, but most importantly, you're losing cells that express PD-1, you're losing cells that are expressing granzyme, and you're losing those effector like CYP3CR1 cells, which are more cytotoxic. And then what my colleague did was something we noticed also in the sequencing was the cell, we had a lot of those CD62L positive precursors in the lymph node, and they too seem to be present in this whole checkpoint response because even though this substantially went down in proportion after checkpoint, this population was still quite well maintained in numbers. So it seems that these cells are expanding and differentiating in this checkpoint response. And as Carlson's paper showing in chronic infection, these cells are important. We want to see how that works in tumors. And we knew from Carlson's work that MYB is co-expressed by these CD62L TPEX, and Lisa showed this was true also within our tumor model, where you can see here in the tumor draining lymph node, CD62L positive cells all are MYB positive. We didn't really see much CD62L or MYB GFP in the tumor, but again, we were seeing these really good cells in the lymph node. And then what she did was made a knockout mouse. So these mice do not have any, they knock out the MYB molecule in all CD8 cells. So we're losing the CD62L TPEX here. And you can see here, these mice also lose the ability to respond to respond to checkpoint, yes. But right now here, what I'm showing you, sorry, is just steady state, but you're showing that with the knockout mice, you have worse tumor control, so they grow out faster because we can't make those effectors. And then you can see here at the bottom that you lose that CD62 positive population in our lymph nodes. So we were losing CD6 T-PEX cells when you knock out MIB. And quite nicely, what she also did was make a bone marrow chimera to make sure that the cells were growing in the same environment and we could see if the changes we were seeing were cell intrinsic. And we also gave this mice checkpoint later on. But as you can see here, that we had a massive expansion of cells in the tumor and also in the lymph node when you normalized it. But this was one of my favorite pieces of data from it, was where we saw that if you treated these mice with checkpoint, the control mice could give rise to an expansion of CYC3CR1 positive cells, but this could not be seen in the MIB knockout mice. So we're really showing that those effectors that were going to the tumor and controlling the tumor were being formed by MIB positive CD61 T-PEX, helping us identify that it was the lymph node T-PEX that were essential to the immune checkpoint response in our models. And just to summarize what I've been talking about, we saw that Hobbit-TRM like T-PEX cells play a very limited role to tumor control, while non-TRM like tumor draining lymph node precursor cells were mediating this response by giving rise to CYC3 cells, and these were the ones that were controlling the tumor. Importantly, Lisa went on to show that these cells required MIB for their presence and persistence, and also to induce that response in after ICP. I haven't talked about it here, but we also went into the molecular mechanisms, and we showed that TGF-beta induces residency and inhibited the stemness in the lymph node as well. And we not just show this in the murine mouse models, but we also went on to show this in human cancer. And just one last picture to show you that immune checkpoint blockade, what we were seeing was, although yes, it can have a limited impact on our T cells within a tumor, where it was really acting on was MIB positive, TCS1 positive precursor cells in the tumor draining lymph node. And then these cells could differentiate into CX3 effectors. And once they got into the tumor, they were the ones allowing us to see tumor control and fueling that anti-cancer immunity. Thank you. Quick acknowledgements to my old lab, my ex-lab, my PhD lab, and they taught me everything